A question as the audience, and that is, could you maybe show me by your hands who of you or how many of you are clinicians? So that would be psychiatrists, psychologists, addiction counselor. Okay, and how many of you are researchers? So that could be pure sort of cell culture, animal studies. Okay, so it's mostly clinical audience. Okay, so we won't go into the nitty-gritty of the studies. Sorry for those of you who are interested. And we'll just go for a quick overview. If you have any questions at any moment, I'll be happy to answer, okay? And I'm actually from Slovakia, just a quick correction, but that's okay. All right, so I'll talk to you today about alcohol use disorder. How many of you know about alcohol use disorder or use it in your, or see it in your clinical practice? Okay, not that many of you, but that's all right. So why are we interested in alcohol use disorder? Well, according to some estimates, it can be actually considered as the most costly and most harmful addiction, at least in the Western world. And that's especially when we consider it not only from the harm to the individual, which is actually shown in this graph in the blue bars, but also from the societal cost. So this include accidents, it might include things such as fetal alcohol spectrum disorder, etc. And as you can see, actually alcohol is by a far margin the most harmful society in most of the Western countries. When we look at just the clinical cost as well, actually it happens to be the third leading burden of disease in North America in terms of disability-adjusted life years. So those are the number of years that you lose from your healthy, functioning life. Now, in terms of a global perspective, alcohol or harmful use of alcohol is responsible for about 4.5% of the global burden of disease. It's responsible for about 5.9 or around 6% of the global deaths. So that adds up to about 3.3 million, or 3 times the population of Antalya, dying unnecessarily every year because of alcohol abuse. And this is especially relevant for younger males. For younger males, where it can be up to 25%, depending on the estimates and the statistics. And alcohol itself is actually causally linked to over 200 different disorders, and this includes cardiovascular disorders, cancers, metabolic disorders, as well as dementias, for example. In many countries, actually, alcohol-related dementias are the second leading causal factor in dementias in the population. And unfortunately, it's quite a complex biological disorder. We don't have a clear sort of receptor where it would bind, and the damage is sort of quite complicated as well. There is a high relapse rate, so we have fairly limited treatment options and we don't fully understand even the mechanism of both the psychosocial interventions as well as the pharmacology. The relapse rate, according to most studies, is actually over 60%, and for most treatment-seeking individuals, it occurs within the first 6 months of starting their abstinence. And it is also chronic multi-generational disorder, so it tends to plague and affect multiple generations in our communities. That is partly due to genetics, because of a 40 to 60% heritability, but also because of cultural and environmental factors which tend to be shared as well. So we don't fully understand alcohol use disorder, but it is an important topic that we should all be excited to talk about. Right? Right. All right. So what does alcohol do on the brain? Why do we care about? What are the structural changes? These are sort of the most easily identified changes, and eventually we'll get also to the functional changes, which we care about mostly. So alcohol itself is neurotoxic, as well as some of its metabolites. Excuse me. And people who abuse alcohol chronically, as well as binge drink, so occasional excessive alcohol consumption, experience a wide whole brain atrophy, which affects mostly the white matter and affects multiple brain regions, such as the limbic system, which is involved in reward, emotions, thalamus, which is sort of the communication center of the brain, hypothalamus, which regulates stress, hormones, basal forebrain, which is involved in memory, cerebellum, and motor coordination, etc. And here is just a clear example for all of you to see. Can you guess which one is alcohol-dependent patient scan and which one is the healthy control of the same age? Any takers? No? All right. So the top image is a healthy control, and at the bottom we have the same age treatment-seeking, recently detoxified patient who's been drinking about 16 to 20 standard units of alcohol per day. As you can see, where's the pointer? Oh, that's not the one I wanted. All right. As you can see, there is enlarged ventricles, which are quite obvious, as well as the cerebellum is quite atrophied as well, which you don't need some fancy statistics to observe it. It's quite obvious based on the sort of the gross morphology of the brain. So when we actually look at on the microscopic level and do the histological on postmortal brains, what we actually see is a double-digit decrease, even in uncomplicated alcohol-dependent individuals. So again, at the top, you would see sort of the healthy representative images of neurons, and you can see that they are very richly sort of branched out. And at the bottom, we see comparable tissue, uncomplicated alcohol-dependent individuals. So there are fewer cells, again, double-digit loss, as well as there are fewer connections that those cells make. Here is also an evidence from histology. This one is from animal studies, unfortunately, but these black cells are essentially new neurons in the hippocampal region, and alcohol unfortunately inhibits the neurogenesis and the maturation of progenitors' stem cells, neuronal progenital stem cells, and the cells also have fewer connections. Now we know that, for example, in humans, it's estimated that the hippocampus has about 1.7% annual turnover. So it is very important not only that we try not to kill the cells by the toxicity, but that we also keep replacing the cells which are normally turning over. The best paper I have found which sort of explains the mechanism of alcohol damage and subsequent recovery during prolonged abstinence is this paper based on animal studies from Fulton Cruz and Kimberly Nixon. It's essentially several lifetimes worth of researchers' findings summarized in one paper. Here is just an example of what happens during acute ethanol exposure. So when you drink too much, essentially there is an increase in degradation of the cells, and we can see that in the sort of immunohistochemical staining here. So there is inhibition of the neurogenesis, as we mentioned, so the black cells would be the new neurons. We don't see that over here. And here the black cells would be cells which are dying, so there is more cells dying in the alcohol-dependent or alcohol-exposed mice. And again, we can see that even more clearly on this picture of hippocampus. So control, not exposed to alcohol, we don't see a whole lot. Here, during the alcohol binge, we see a lot of sort of black staining. That means there's a lot of cells dying. And this persists even several days into the abstinence. So cells dying, not good. And there is also death not only of the neurons, but also of the supporting glial cells, again, in double-digit percentages. So what causes this? Again, it's quite a complicated process. The three sort of key reasons for the death is oxidative stress, so those would be inflammation and reactive oxygen species, and also excitotoxicity for people who have been chronically abusing the alcohol, because there is sort of an attempt for the body to establish a homeostasis, and as you know, alcohol is a depressant, so you have too much excitation in the brain. There is also toxic metabolites of alcohol, such as acetaldehyde, aldehyde metabolites, which are potent mutagenic, carcinogenic compounds. And there is also a decrease in the sort of pro-survival, pro-repair, pro-function factors such as BD and F, which we can actually even measure in the plasma and the cerebral spinal fluid of patients. So it's not just in the animals. And on the other hand, what happens when we stop drinking? We see a massive proliferation and new neurogenesis, at least in the animal models. So, as you can see, there is about 3.5 times increase in the protoneuron progenitor cells, and after about 1 month, these differentiate to about twice the rate of new neurons being produced. So here in the black, you would see the alcohol not exposed mice, and here you would see in black sort of the new cells in the recovering alcohol previously exposed mice. Again, here is another slide which in black shows essentially increased cell activity, increased repair, increased regeneration. In the healthy control, we see very limited sort of black staining. In the immediately after the binge, we don't see all that, but after prolonged abstinence, or 3 days of abstinence, we see a massive increase in the proliferation and the repair of the cells. So this would be sort of the whole image. In the red, we see the degeneration, which is associated with the acute toxicity. And that's why even from the most recent issue of JAMA Psychiatry, Dr. Nutt's paper, which was talking about how decreasing, even if your patients still continue to drink, decreasing the amount of alcohol that they drink is very important, because that's the acute toxicity. The higher the concentration, the more of this damage happens. And then after prolonged abstinence, we have a spur in new cell growth and new differentiation of neurons, as well as the supportive cells. So what about humans? We have essentially all the evidence we want, but it's not very direct, because unfortunately, because of ethical and practical reasons, we can't really connect longitudinal biopsies from people's brains. I don't think there would be too many volunteers, and it would be obviously very unethical. So what do we know from the non-invasive studies? We know that there is a partial gray and white matter volume recovery. And the recovery is nonlinear, and it depends on the brain region. Some of the damage is not reversed, and it's especially relevant for people who resume drinking again, because there they don't actually recover from the baseline according to the results. Here is just an example from one study. So you see the ventricle sort of shrinking, so the brain around them filling back in the areas which were damaged after about 2 months of abstinence. Here is hippocampal volume increase after only about 2 weeks. We see in the sort of the darker bars, those would be the healthy control participants who don't abuse alcohol. And in the lighter blue, those would be the patients essentially when they're stopping drinking and after 2 weeks of supervised abstinence. So that's just what we were looking at, the subregions of hippocampus. And we see similar story also with the white matter, where prolonged abstinence results in increased volume as well as increased integrity of the white matter microstructure. Unfortunately, there is also a selection bias where the people who survive, who remain abstinent, tend to also have better results beforehand. And that's a problem that has plagued many studies, because a lot of the studies are only cross-sectional. So we don't have a longitudinal follow-up. That's a problem to this day. So, in summary, there is a solid structural evidence of brain plasticity during alcohol use disorder, recovery from both animals as well as human studies. How does that impact sort of behavioral and functional changes? What is the fMRI evidence? Well, in general, for all addiction is associated with a strong bottom-up appetitive drive. That's sort of what drives the intoxication, the disinhibition, and there is a weaker top-down executive control. So that essentially results in you ignoring your day-to-day activities, and drinking despite of knowing that it's bad for you. There is numerous ways and numerous models of addiction, but they generally have these three key components. So they would have the reward circuit, or the reward stimulant, and that would be based around dopamine networks in ventral tegmental area and nucleus accumbens. There is a memory component, which can also have a positive or negative emotional attachment, and that's centered around amygdala and hippocampus, so that's also important, for example, for withdrawal. And then we also have regulation of emotion executive function. That would be mostly your frontal regions and anterior cingulate cortex. So this is an example, and as you can see, a lot of the regions sort of overlap. In blue here, we have the binge intoxication area, so a lot of the deeper tissues and the basal ganglia. We have the preoccupation anticipation, so that would be the memory as well as the executive function areas, and withdrawal negative effect, again, overlap, plus the amygdala, for example. There is another look here. These are in red would be the executive control network. So those would be the red regions, and blue regions, those would be the appetitive or the reward drive regions. And as you can see in purple, there is a lot of overlap between those regions. So when we look at resting state fMRI, how many of you know how fMRI works? We're essentially looking at changes in the oxygenation level in the brain, from oxyhemoglobin to deoxyhemoglobin, and that's correlated to brain activity. More brain activity, more metabolism, less oxygenation of the hemoglobin, and hemoglobin in its oxygenated and deoxygenated form has different magnetic properties, and that's how we can detect it on the scan. So in the red, we usually see those areas which are lighting up. Those would be the areas which are metabolically active. And we can replicate this very well in EEG, fMRI, etc. And it describes a lot of the regions which seem to have a correlated sort of activity. So this would be just an example. So in the blue and the red line, here we have, I guess, cerebellum and hippocampus. So these two would be highly correlated, so they would show up as a network on the resting fMRI. Here we see they're anti-correlated or they're sort of disjointed, and they would not show up. They would have low synchrony or low functional connectivity, and in general that's considered to be bad. So you want high functional connectivity and purely sort of nicely narrowly defined networks. If you see diffuse networks or if they're sort of not showing up, then that's bad. All right? So how does this relate to alcohol use disorder? So we have generally a broad decrease in synchrony across most of the brain networks. The most important ones from sort of the mechanism and most of the models of addiction would be the salience networks. So they involve a lot of the anterior cingulate cortex as well as insula. How many of you have heard about the nicotine study and sort of insula? How if you have an ablation to insula, some people have been reported to just quit craving cigarettes. I mean, that's very interesting, right, for addiction researchers. Then we have executive control networks. That's a lot of the frontal regions. And then we have the basal ganglia, which are associated with some of the reward areas, as well as sort of the network communication hubs in the brain. So the literature is quite sparse. There is sort of poor homogeneity of the samples. A lot of the patients have multiple comorbidities. There is a lot of cross-sectional studies, not a lot of follow-up studies, and smaller sample sizes. Nonetheless, in general, there is evidence of increased sort of synchrony between ventral striatum and other components of the reward network. We also see that some of the studies report sort of compensatory recruitment of parallel brain networks to help achieve the same level of performance on cognitive tasks, even at, for example, slower speed. And here is just an example of the cerebellar cortical networks, where patients will actually recruit the parallel network next to it to compensate for the damage of the primary network that we see being used in the patients, in the healthy controls. So in summary, alcohol is associated with widespread functional dysfunction, sort of the poor homogeneity and poor functional connectivity. There is maladaptive circuit reinforcement, especially the reward circuits, as we have mentioned. And there is also compensatory recruitment of parallel loops to help achieve the same level of cognitive performance. So when we look at abstinence, what happens? When we look at cognitive and behavioral performance and recovery, we see there is selective functional improvement within aspects such as nonverbal memory. Some functional improvement takes years to accomplish, and this can include general memory, psychomotor speed, and some aspects such as static balance can be impaired for a long time, sometimes indefinitely. In general, we call it decades. So there is a clear evidence from testing and even from clinical practice that there is some level of plasticity and functional improvement in the patients. Now the question is, how does this happen? Here is just an evidence, again, of dopamine receptor availability recovery. At the top, we have crystal meth addicted individuals. So these would be the controls. That's what we would normally expect. Here is an addicted person to methamphetamine early in the recovery and after prolonged recovery of over 1 year. And as you can see, the recovery has been quite substantial. Unfortunately, in alcohol, there is still recovery, but as I mentioned, the etiology of the disease and the etiology of the recovery is quite complex, so it is not such a clear sort of improvement as we might expect in some of the other addictions. So what does the resting fMRI literature tells us? Generally, the recovery is a gradual process. there are a lot of adaptive changes. So we don't go back to how we were before, but we have an overcompensatory sort of adaptive state to the new abstinence state. So we have an increased executive and decreased circuit activation, even compared to the controls. So most of these studies rely on region of interest analyses. So these would have seed regions. For instance, here we have the nucleus accumbens, which is a very important in the dopaminergic sort of reward signaling. And we also have executive control network, which would be the subgenual anterior cingulate cortex over here. And this one is important in sort of all sorts of executive function things. And when we see the voxels which sort of activate and deactivate at the same time, this is just an example of what the nucleus accumbens activation network would be in the long-term abstinent alcoholic individuals, and this would be in the healthy controls. Here is the same thing with the subgenual. Sorry, I guess we're moving to the next slide. So what happens with the prolonged abstinence? So in this case, the graphs in the blue, that would be the healthy controls. In the green, these would be the short-term abstinent. And in this case, this would be a few months, around 3 months of abstinence. And in the sort of the khaki color, you would have the long-term abstinent. In here, it was around 7 years, I believe. And what we see in the executive sort of top-down changes is that there is increased synchrony from both seed regions in the dorsolateral prefrontal cortex. So those would be these blue blobs over here from both of the seed regions of the reward as well as the executive function. So we see sort of a continued trend where there is increased activity, which might be linked, we think, to shifting performance abstinence and inhibitory control, which are all very important for maintaining abstinence. Now, on the other hand, we see the opposite effect in the bottom-up changes in a lot of the deep sort of reward-related tissues in the limbic system, such as the caudate, the thalamus, etc. So again, in the blue, we see the healthy controls which do not abuse alcohol. In the green, we see the short-term abstinent individuals. And here in the khaki color, we see the who've been abstinent for over 7 years. And again, there is a clear trend downwards. So what does this mean? That we don't really go back to the pre-disease state, but we overcompensate. We have to have a lot lower sort of activation of the reward areas to be able to cope, and a lot higher activation in the control, executive control areas to be able to prevent ourselves from, relapsing, starting to drink again. There is also a relation in this data to prediction of future relapse. So there is only emerging evidence so far, but we see that the people who relapse in the future, so looking retrospectively at the data, we actually see that most of them have poorer synchrony in the data. They have lower activation levels, etc. So these overcompensatory changes don't happen as much in those people who then end up relapsing later on. And this also correlates with severity, length of ethanol abuse, as well as the age. And so these are some of the networks where there has been a demonstrated significant correlation for any of the researchers. So again, what we would expect, reward, executive control, and some other ones like the visual network and I guess the salience network, which involves insula, for example. So again, we see the similar trend. So the people who remain abstinent, they have increased sort of overcompensation versus those who relapse have a lesser overcompensation. And we see that across multiple brain regions. So this is quite important when we work with clients and clinical patients, because if we can predict even before, they might relapse, that they are much more likely to relapse, it's not necessarily their fault. It is essentially the biology of their brain which might predispose them, which might make them more likely to relapse and much more difficult to maintain abstinence. And that also opens up new possibilities for treatment. So one such possibility is brain modulation and neurofeedback. So brain modulation is essentially using magnetic stimulation or direct current stimulation to essentially change how the brain functions. And neurofeedback would be similar to that, but you're sort of using the person to help adjust how their brain signal appears. So this type of research has been going on for over 40 years, but it's not very high quality research. There is a lot of case studies. The early research was essentially similar just to meditation, so it doesn't really have a lot of substantial scientific merit behind it because we cannot really quantify what they were measuring. Sorry, it keeps changing on its own. So the other technique is deep brain stimulation, and this is also very sparsely documented. It's mostly in case studies with other comorbidities because it is an invasive procedure. But unlike some of the other invasive procedures, it is reversible. There is some gliosis next to the electrode, but you're not slicing anything out, right? You can always remove the electrode from the brain. And sort of the most promising recently technique is the fMRI and EEG neurofeedback. Now, the fMRI is quite interesting, especially to researchers, because it's not very practical for the clinical practice or for the individual to do at home. But the EEG is actually quite an interesting but very early technique, which could potentially have a lot of use for especially the long-term sort of treatment and therapeutic contact. The regions which have been studied in the past is dorsolateral prefrontal cortex, which we have seen previously in some of the fMRI studies, anterior cingulate cortex, as well as the nucleus accumbens for some of the deep brain stimulation studies. Now, the evidence suggests that there might be potentially decrease in craving in some of these studies, but these are only with few samples, with smaller N numbers, and there is also potential increase in inhibitory network activation, and some studies also claim decreased reward network activation. The EEG apparatus here is very simple. It would have to be a little bit more complicated so that we can actually localize it a little bit better if we were to do a translational research. So there is a lot of promise in these techniques, but we need better quality research. We need to study different brain areas to make sure we are targeting the right approaches, and to make sure that there is actually merit and clinical efficacy behind these techniques. And there is some large sort of multi-site studies which are going on. This is a study from Germany, which they're trying to do from 2018 onwards, where they plan to recruit about 100 participants for fMRI neurofeedback using 5 different study groups in their design. So, conclusion. When you're suffering from addiction, it's kind of like driving like Mr. Bean. You're not fully in control because your brake pedal and your gas pedal aren't really working. When you're actively addicted, your brake doesn't work quite well and your pedal for the gas is super sensitive. So to recover from it, unfortunately, you have to have the opposite, and that's what sort of happens according to the fMRI studies of brain plasticity. All right, thank you very much for your attention. These are some articles you can look up if you're interested. This is my contact information, and if you're ever interested in alcohol research, there is some shrews in Malaysia which are constantly drunk, and somehow they survive. All right, thank you very much.
Thank you, Michael.